Hi Ron, how are you doing?
Hi, Ben. Nice to see you again.
Yeah, we definitely haven't been doing this for about an hour and a half, with technical difficulties or trying to sort things out, have we?
Yes. And in
Yeah.
between, I've been checking on my son, Whitney.
I'm checking. Yeah, so I'd like to say thank you to yourself again for doing this, and also to Janet for sorting out the technical issue. She managed to sort the technical issue out, and yeah, so we've managed to do it. So thank you for fitting everything in and doing this casual chat like last time with me. I really appreciate it. So yeah, thank you. But so I just wanted to, I've got some questions mainly about the symposium and the research and various things. And I've also got some patient questions this time. Um, but you can just go wild with them and talk as much as you want or talk as little as you want with them. So I guess to start it off, I just wanted to talk about the symposium. So obviously we had the OMF third annual symposium.
Yes,
Uh, that was early September, September the 7th, I think, wasn't it?
right.
And I just wanted your thoughts on how it all went, really, just general just general thoughts, any signs that were different to last year, etc., like that.
Well, in general, I think we're making progress. We had some new people give talks. Um, I did bring in some engineers to work on some of the problems, and now we're trying to sort out the differences that they find versus the differences that we found earlier. And so we're in the midst of exploring that. Earlier, we found a deformability, and the new device doesn't see it. So we're looking at the differences that were present. One of our concern is that they're putting the red cells into phosphate buffered saline, and it may have to stay in the plasma. So they're setting up new experiments to see if that will make a difference. We're looking at just the details. Sometimes with these techniques, very subtle things that you do make it work or not work, and you don't actually know why.
Yeah,
So it always is a difficult time to transfer the technology. I've had a lot of experience that, and I've also a lot of companies have experience um,
I
when they
think.
transfer the technology and they try to give everything and all the details, but there are certain things that change,
Mhm.
and then all of a sudden doesn't work. And
Yeah,
that's just
think that.
such a common problem. Uh, but I've told the community that when we have these symposium, we're going to give you the very latest up-to-date things that we're doing. This
Mhm.
is what's happening in the lab right now, and sort of full disclosure. And we're not waiting until we have a publication out and then we can tell you about the publication. Um, in my experience doing these complex things, it usually takes 2 to 3 years before, when you start something, before you actually have the paper out. My feeling is that's just way too long. And NIH likes the papers. I do too, but I think we can communicate things much earlier than that.
Yeah, I remember you, I think, saying last time, or maybe when we've talked off, like when it's not been recorded, about how, um, I mean, it's quite obvious when you think about it, but it's one of those things that sometimes you do have to think about. It is like, for example, the media that or like the medium, sorry, that the cells are in can really affect the results of the experiments, and sometimes things can be assumed about the medium, like whatever's added in as a buffer, etc., or anything like that. Um, and that may have changed from one year to the next, and that can actually, obviously, like you're talking about, can affect the results. And it's quite a hard thing to pinpoint down because it's obviously something that could go unnoticed if you weren't paying close attention.
So if we actually do develop a technology that we want to export, we often do a lot of troubleshooting. We look at changes in the protocol, changes in the concentrations to see if there's anything that's very sensitive to the technology. We
Yes.
haven't begun to do that with the metabolic trap, and I'm sorry, not the nanoneedle electroimpedance.
Yeah.
And we're worried that it probably does have some very high sensitivities. Um, but we'll figure that out.
Okay.
The problem is you may pick a condition that is just right at the edge of having it work. And if you make up the solutions a little incorrect, then it doesn't work. And
Yeah.
we want to find out if that's going to be the case. But right now, it's not ready for export. But we're working on trying to get it so at least it could be exported to the scientific community. That would be the first pass because it'll be easier for them to take on something technically quite difficult. And
Yeah.
then later you go to testing laboratories, and they usually use fairly inexperienced people, and it has to be very routine and simple.
Yes. I mean, that actually follows on quite nicely because by far and away, I, I'll ask the questions like from the other patients at the end, but by far and away the biggest question, by just by a magnitude of I don't know what was basically what's in the blood or what isn't in the blood or what multiple things are in the blood to cause this impedance signal. I think people and obviously myself and everyone is because it sounds very simple. It sounds like a very simple thing to, something's in the blood. What's causing
Yeah.
this thing? It's obviously incredibly kind of symbolic in a way because I think there's been some diagrams going around on Twitter that say like ME cells in healthy patients' blood and they're okay, and then you have. Hope I've got this the right way around with my cognition, but then you've got obviously healthy cells in ME patient blood, and they have that same or very similar impedance measurement. I don't know if you can clarify.
Well, that's actually fairly interesting because what it means is if we have the cells in from a patient and then take the cells out and put it into healthy plasma,
Mhm.
we lose the signal. So that means that that the plasma from the patients have to be in contact with the cell at all times. So
Yes.
it's not like you treat the cells and now they're stuck in this pattern. They go out of that pattern very quickly, which raises some of the issues with people using the seahorse instrument and trying to compare it, because the seahorse instrument, how it's routinely done, is not done in plasma, it's done in buffer. And
Yeah.
so they may not see what we're seeing because of that. We've tried to run it in very small amount of plasma, but we haven't gotten to be, that hasn't been successful.
Okay.
So that's why they run it in buffer.
Can you just give I guess a brief kind of explanation of why it's not quite so simple, that it's just something in the blood, why it's quite hard to find out what that may be or what those things may be?
I think we can figure it out. It's just very difficult with our current setup because we have a $30,000 instrument that can only do 2 samples at a time. And
Okay.
so, and it takes several hours to do the setup and run. And what you would be doing is fractionating the plasma with different techniques. And now you would say fractionate it into 10 fractions, but now you got to measure all 10 fractions. So the
Yeah.
problem is, well, we could do 2 of the fractions, and then we could get a patient to come in the next day because we shouldn't ask the same patient to get blood. And so we have a different patient to do another 2 fractions. And how do we interpret that? It's just not the way to do this experiment. And it would take a lot of time and effort. The way to do this is to change the electronics and the way the chips are also all done and be able to do 100 at a time.
So it's
And it doesn't take much blood for this. One patient donation would be able to do 100 samples. And so now you could do the fractionations and in fact do quite a few different fractionations and run it all at the same time. So that's a much better way to figure this out. And
it's a kind of a funding and scalability issue, which are kind of intrinsically linked, obviously. But
well, we have a grant in, and we'll hear this month for Raheem whether that grant will be funded. I'm
okay.
pretty optimistic that it will because he's a young investigator, and there's
Hmm.
given preference for that. But the proposal is pretty complicated in the sense that he has to rebuild these electronics and make a very high throughput device.
Hmm.
And I just don't know if they can find the right reviewers for this.
Okay, that's interesting. That's obviously, yeah, Rahim is obviously the person he's the engineer behind the red blood cell impedance device, isn't he?
No. No, it's the
No, have I got that completely wrong?
it's a different group. That's the work that was initially started at San Jose State, and that's continuing.
But so Rahim, so the initial impedance device was that not constructed by Rahim?
No.
Oh, okay.
Which device are you referring to?
The
The original nanoneedle device?
yes, nanoneedle. Yeah,
That was invented by Raheem
yeah,
and
that's yeah,
fabricated by him.
yeah,
He's now moved to the University of California at Irvine. It's a
yeah.
short plane flight. He does come up almost once a week. Um, but he just delivered on Friday a new batch of chips. That was his prime start at the new location of
Okay.
getting a new graduate student, getting a graduate student trained to fabricate. We haven't yet tested them, so I'm hoping they will work.
Yeah.
If they don't, then they have to start over and make another batch. But
Yes.
that new batch will help us do a lot of experiments, and we'll do the ones where we can get a good experiment just using one or two chips
Yes.
with our current device. And there's a number of things that we can do.
Okay. That makes sense. I thought I was going mad there for a second. I'm glad that yeah, that makes sense now. Yeah, okay. So another thing that was, I think this was with from Uppsala, the center there that you guys are working with, because now you've obviously got Stanford, you've got Harvard, and you've got Uppsala University.
Yes.
So you've got three centers which are working together, which I think is quite incredible, really, and a huge step forwards. And one of the things I picked up from that was really interesting, um, that I think it was them that were doing this, which was the proteomics of neuroinflammation markers in patients, autoantibodies, autoimmunity, and steroid and thyroid hormone regulation.
Yeah.
They're, I think they're also doing microglia imaging. I'm reading this off my little notes list.
Yeah, he gave the talk, but he didn't interact with us. He was out of the country, so he didn't
Yeah.
come to our working group.
Oh, okay.
And we do have to talk with him because he's doing some things that we're also doing. And we just don't want duplication of effort. Validation of results is valuable. So it has to be along those lines. And but also, we haven't done any spinal fluid, and he's an expert in that as well. So
Yeah.
there are a number of experiments that I think should be done. We'll either ship the technology to him, or he will ship samples to us. Probably the shipping to us will work because most of these, I'd like to look for any presence of any viruses or bacteria
Yeah.
in the spinal fluid. And we would do that by looking for DNA. DNA is very stable, it can be frozen, it's not a problem.
Hmm.
So we'll send in freezing canisters, and he will just drop the spinal fluid in, and then we'll assay it.
Okay. Will will the freezing of, I mean, obviously it will in some way, but will for the, I'm presuming obviously for the purpose of what you want it for, the freezing of the samples won't affect the results in terms of, because obviously I know for certain things you need fresh plasma.
Yes, if we're doing something sort of biological, which the
Mm,
nanomillimeter is a biological assay,
okay, yeah,
the freezing can be a real problem. We haven't gotten any good results with freezing. You still get a weak signal, but it's not trustworthy. And
okay,
so we don't want to use frozen samples. But DNA analysis is, we have a lot of
yeah,
experience. Not the world has a lot of experience, not just us, uh,
yeah,
about handling DNA. DNA is very stable, but RNA is also stable. So we will look at the RNA and the DNA.
yeah,
It's just something that needs to be done. We have the technology to readily do it. So on that front, we're setting up to do RNA analysis. So we can look at the RNA viruses.
okay,
And that's a project that isn't funded. In fact, none of the stuff that Pidong Shen has been doing with DNA analysis and looking for viruses um, is a specific project that's funded.
mm-hmm.
Um, and this is a good time to make donations because Open Medicine Foundation is now having their Triple Tuesday. And that any amount of donated will be tripled. And so that makes your donation more effective. I wish
Yeah,
research weren't so expensive, but it is. And we are putting in another grant on the 11th of November. Um, and we're going to have at least one or two grants every NIH round.
okay.
And the best way, if they don't want us to keep submitting grants, is to give us a few.
Yeah, I mean, again, yeah, it's Triple Tuesday at the moment, so obviously every if someone gives $1, OMF get $3. If someone gives $5, OMF get 15, etc., and so on.
Right.
And I think the aim this time is to raise $1 million, which is obviously um,
Yes.
a lot of money. But it's also at the same time it's quite easy, I think, to forget that so much work so far has been self-funded or funded by OMF
That's
at
right.
Stanford. And I was looking on their website the other day. Obviously, I'm a patient advocate and a volunteer, but I'm a severe patient first and foremost. So I'm interested in the organization and how much money people are raising and how much money that organization is raising. And I think it said something close to 20 million in 5 years, maybe. Um,
Yeah.
which I think is quite an astounding amount of money, and it's enabled you guys and other now the other Harvard and Uppsala to be able to continue researching. So with Triple Tuesday obviously being a focus at the moment, that money goes so much further. Um, and I guess we're very fortunate in to have those donors who are willing to match and provide those extra funds. Um, so hopefully, if people support the cause, they'll donate, and we'll get there faster until NIH pull their finger out, basically. So yes, um,
We have we're setting up a new collaboration, which is an area that I've been wanting to get into, and that is he's a neurologist at Stanford.
okay,
And why he's particularly of interest to us, he has a lot of experience with uh, the effect of plasma and, materials in the plasma that affect the brain. And
okay, wow,
so, since we see effects of plasma, uh, we would like to try to explore that. And he's very interested in doing it. We don't really have a budget for that. We don't know how expensive it's going to be. Uh, but that hopefully will be another project that could be very beneficial. We know
yeah,
there's a lot of neurological effects, and it
hmm,
would be wonderful if some of that could be modified by things in the plasma. So it's either something bad in the plasma that's making things worse, or it could be the absence of something good, and both of those we will look at the approach for that.
That leads really nicely on to actually one of the things I'd noted down, which is one of the compounds that was really low in patients. I hope I'm pronouncing this right. I have read about this compound before, but it's indolylpropionate. Is that
Indolylpropionate? Yes.
indolylpropionate?
It's not a very complicated compound. You can actually buy it, but it's not clear that it's safe to take because it's made by a chemical manufacturer.
Yeah.
And there is a company that's exploring that as a supplement for brain protection. It's in clinical trials at the moment. The major thing we need to find out is whether the preparations they've made are safe. And if it is, then we would like to try to do a collaboration with them. We haven't talked to them yet. They have to get results before we can do that. But we're very willing to work with industry as well as academics to come up with some solutions.
Yeah. I mean, reading about it was very interesting because obviously, just as a bit of background, if people aren't familiar with that compound, it's a neuroprotective made by a species of Clostridium bacteria in the gut, isn't it?
Yes.
And so with all the gut issues we have and some of the findings, I think, by Hansen, and just, I mean, there's a lot of findings now, evidence of dysbiosis and that various things like that. I think it's a very interesting compound in itself, and I think it's one, I might be wrong on this, and you can correct me, but I think it's a compound similar to melatonin in the sense that I think it's an antioxidant, but I don't think it's first of all a pro-oxidant. I think it's just like an antioxidant, like an innate and it functions as an innate antioxidant.
Well, yes, it could be. I don't know. We don't know its biological effects.
Yeah.
But it could explain why fecal transplants are helping people.
Yes.
Because all of the severe patients had low levels.
Yeah.
And by putting the organism back in, but the fecal transplant is going to have to be done correctly because if it's a Clostridium, it's killed by oxygen. So I can imagine sometime it not working because you have exposed the transplant to too much oxygen. So it might be simpler and cheaper to simply get the compound and take it as a pill. Since it's made by the bacteria in the gut, putting it back, the compound in the gut should be fine. And it presumably isn't going to get destroyed because it's not destroyed already. So that's going to take a while before that's present, but we are working on that. So right now we're putting a little bit of a heavy focus on kind of trying to come up with some ideas that could help the patients as we try to figure out what's causing the disease.
Yeah. Okay. That's really interesting. It's one that really kind of stood out to me. I know it had been mentioned briefly before, but it was mentioned again, and that I find that personally, I find that very interesting with having a lot of gut issues myself and talking to other patients. I find that one very interesting. Another thing that was very I mean, it's a big thing recently, I think, because of Jem Breyer, a guy called Jeff, who has a website called Mechanical Basis, and also another patient called Matty, is CCI, so craniocervical instability, which I'm going to do a very bad job of explaining because of having a foggy brain, but it's essentially the laxity of ligaments and pathology of basically connective tissue and ligaments affecting the brain stem and various organs in that area. That's not the best explanation at all, but I think a lot of people know about it already, and it's obviously if the brainstem is compressed, various, yeah, I mean that's gonna be truly not very good for obvious reasons, but having heard of these like success stories that Jen Breyer has had, and um, um, Jeff and Matty, I just wonder what your thoughts on CCI are. Obviously, as from a patient perspective, I kind of want I mean, these stories are quite incredible to me. I don't I don't understand in terms of there's some theories out there, and I know Jen Breyer is working on some, and some have been discussed on forums before on why so the issues have been alleviated for some people who have had surgery. It's a small number at the moment, but it's fascinating to me that someone diagnosed with ICC, ME/CFS, fulfilling all of the criteria, having essentially a structural issue, resolving most of their symptoms for the two people I know, and I think the other person is resolved. They've gone from severe to moderate, which is a huge step up. I just wondered your thoughts on if what your thoughts on CCI are, if you're investigating it, and yeah, just your general ponderings, Ron.
Well, I've talked to a number of physicians that specialize in ME/CFS,
Hmm.
and their thoughts are that it's probably a fraction of the patients may have that problem, but it's probably not all. And so that's one of the confounding issues. Then, if it's not all, then why do the two populations seem to be exactly the same?
Yeah.
And so that's a bit of unexplained. One of the things that is also consistent with the CCI is that we find that in the severe patients, some of the highest metabolites in the blood are lysine and hydroxyproline. Those are major constituents of the connective tissue, the
Hmm.
collagen.
Yeah.
And so that would be consistent with a degradation of the collagen. That's not proof that that's the case, because it could be almost the opposite, that because the collagen is weak, the body is actually revving up the production of those molecules so that
Yes.
they can be used in new synthesis.
Hmm.
So, however, it just says that that is consistent with connective tissue issues because
Hmm.
we see the building blocks higher. We have to sort that out. We have some looking at, we're now doing a lot of pathway analysis. Rob Fair is really good at that.
Yeah.
And we're measuring a number of things in the pathways, and are finding places where we could stimulate the degradation or inhibit the synthesis. And that's going to be explored a lot more. So we do have a pretty big effort on pathway analysis. And we're trying to get all the different components in the pathways to be quantitated. Um, that's going to be expensive because many of them, nobody does it. And so we're going to have to set up special tools to be able to do that.
Yeah. 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 I mean, you literally answered my next question, was the link about EDS, hydroxyproline excretion being high and possibly indicating the degradation of collagen. Um, so that's really helpful. I don't know if this is still the case, but I did read, I think it popped up somewhere that you guys were looking for people before the people who were potentially having CCI surgery,
Yes,
and then after for blood samples. Is that still
right.
the case?
And we'll do that. We'll look at the metabolomics. We'll get their sequence, and we'll do the nanoneedle assays. I don't know the timing of that. We have found a few patients. Some of them are, you know, not local, and so getting their sample may be a little tricky.
Yeah.
Um, but we are capable of traveling to patients. We've done that a couple of times,
Hmm.
where we've actually gone to patients. And we have most of the equipment that we need to process a bit portable.
Yeah.
Thinking of equipping a van um,
Okay.
with all the equipment that we need so that we can actually go to patients that are unable to come to us.
That
Because
sounds
otherwise, we're
fantastic.
just looking at the local population only.
Yeah, that sounds fantastic. Very cool. Equipping a van. Um, it's appealing to my geek side. Um,
Right.
yeah. So okay. So is that just to clarify? Is that still ongoing? Are you still looking for potential PCI patients? Is that something that if someone's having that operation for whatever reason, you would want to be in contact with them?
Yes.
Okay. I just
Especially
wanted
if
to.
they're, the ones doing this in Europe are going to be much more difficult.
Yeah.
But if, the closer they are to us, the less expensive it will be.
Yeah.
Sometimes those patients actually come to the Bay Area because we have a number of physicians that specialize in ME/CFS. So
Yeah.
um, they come here to see the doctor and maybe before they fly to New York.
Yes. Yeah, that makes sense. Okay. That's that's good. Um, so what was I going to say next? Mark Davis and T cells. So that was one thing that was in the symposium, and patients were also came up a lot in for patient questions. Basically, people wanted to know the progress, but obviously, I think in the symposium you mentioned, it was mentioned, sorry, that the immune T cells were reacting, but you've now since found out they weren't reacting any more than control patient cells were, but you don't know whether they're reacting to something different, and that's what you're trying to find out next. I don't know if that's
Yeah, and Mark Davis had a long-term commitment and he couldn't change. Um,
okay.
and so he wasn't able to come to be at the symposium. The work isn't complete, but they are making, my understanding, they're making some progress in that area to find out
Hmm.
what they're reacting to. Um,
Okay.
the initial thing that started this whole project is we had a fairly small number of patients and controls. And the patients had a lot more reaction than the healthy controls. Since then, we've done a lot of patients and a lot of healthy controls. And now the difference doesn't look so great. We find some healthy controls that have a lot of reaction. Maybe they just have had a cold or something in the recent past. And we've also found patients that have almost no T cell reactivity.
Okay.
So the level of reactivity is not going to be a good indicator. So the question then is, that's why we have Mark working on this project to look at what they're reacting to. It's
Hmm.
very important to try to figure out if there is a pathogen.
Yeah.
Because in most cases, if there is a pathogen, we know how to get rid of it.
Yes.
And then we know how to assay it to see if it is gone. So that's why it's so important to keep searching for a pathogen. Um, all the indicators would be consistent with a presence of a persistent pathogen. Uh, uh, just to name one, the disease sleeping sickness in Africa. If you look at the symptoms that the patients have, they're pretty indistinguishable from ME/CFS.
Hmm.
And they call it sleeping sickness because they switch their day-night cycle, which a lot of patients do as well. And
Yeah.
we have looked for, and it's a trypanosome, and we have now searched a number of samples for trypanosomes. Um, and we're doing a broad search for trypanosomes, all the ones that have been discovered so far, looking for things that are conserved sequences in them so that even if it were an unrelated trypanosome, we probably would see some level of reactivity. And we don't just do one probe, we do 8 to 14 different probes for each organism. So all we need is one of them to work. So it's a pretty good assay. However, it may not be a trypanosome.
Yes, I mean, one thing that's often brought up and is I'm really interested in too is enteroviruses and things like Coxsackie that may harbour themselves in the gut and various things like that. But again, if you're doing RNA analysis, that should be taken care of.
Well, it should be. It's a little more complicated because RNA is not nearly as stable as DNA.
Yeah.
So it's not going to hang around for nearly as long, and we'll have to develop some good, healthy, some good controls on that. There is some experience in it, so it's not without any experience.
No.
But you certainly can find that when someone has an infection with those viruses, so we'll do all those kind of controls to make sure that we actually can see that. We're also, we also take seriously when patients write into us, what about this or what about that? Of course, many patients have worried about the enteroviruses, and we certainly take that seriously. That needs to be looked at. It's just the technicality of doing a good job of it. And
yes.
Also that there has been a patient that has mentioned possibility of a nematode. And that's a bit of a tough one because that's very difficult to get the DNA out. But we know how to do that, I think, and we will probably explore that as well. Anything that's kind of as an odd organism could be the cause of this.
Yeah, no, that makes sense. That's good to hear.
Other people have looked for, for infectious diseases, and I'm sure that Ian Lipkin has done a lot of searching, coming up negative. We really need to publish the negative results somehow because it's important to know what you've looked for and haven't found.
Yeah, and the methods involved as well.
It's just so important to make sure that that's not the cause, because you could probably cure it if you figure that out.
Yeah.
And if you couldn't, it would put a big driving force for industry to develop drugs that would kill those organisms.
Yeah. Yeah. I think it was also mentioned that you were testing for mycotoxins and mold. I think you might know a guy called Eric Johnson.
Yes.
He's fairly.
He's visited here. We've had people go to his to visit him. We haven't started anything. I gotta find the right people. Sometimes you can't just start a project. You really have to have somebody that's really an expert in that area, because you don't want to reinvent all the different methods and so forth that are out there. It's a lot cheaper and faster if you can find the right person. So we've been looking for the right person for doing that. And that's a project that needs funding as well.
Yeah, I was going to say it.
And it can't just depend on NIH, because they will not fund anything that you haven't already done it. So
yes.
It creates a huge problem when you want to start something new. There just is no mechanism for doing it in NIH. That's why people, when they start a research program, often do that same level of research their entire career, because it's so difficult to switch.
Yeah.